Good afternoon, everyone. May I please have your attention? Welcome to the session, Immune-Mediated and Related Disorders. My name is Marina Dobrovolskaia. I will be your session chair for this afternoon, and I'm also your first speaker. And I'm going to talk about methodologies for assessing nanoparticle immunotoxicities. This is an outline of my presentation. The main question that I would like to address is whether or not there is an ideal method or model to study nanoparticle immunotoxicity. So please remember this question. We'll come back to it at the end. It is very well established now that nanoparticle physicochemical properties determine their interaction with the immune system. An assessment of nanoparticle effects on the immune cells and organs became an integral part in the translational studies on the journey of translating these materials from bench to the clinic. At the Nanotechnology Characterization Lab, we developed a comprehensive assay cascade, which now contains over 100 assays to look at physicochemical and biological properties of nanomaterials. This is a summary of our immunology assay cascade. As you see, we are using a lot of traditional in vitro and in vivo methods. Luckily for us, many assays in our immunology assay cascade have very good in vitro in vivo correlation, but nothing in this life is ideal. These assays are not ideal, and often we have to look around for alternative models to answer specific questions about nanoparticles' immunocompatibility. Each nanoparticle product is unique, but similar to how everyone in this room has unique signature, nanoparticle-based products have unique immunological signatures. And some similarities or trends exist. Two of them I share with you on this slide, and they include chemokine induction by lipid and polymer-based nanoparticles, and type 1 interferon induction by nucleic acid-containing or nucleic acid-based nanoparticles, and especially nanomaterials that contain RNA-induced type 1 interferon. I like often to think about nanoparticles as VIPs. Why do I say so? We all know that sometimes it is very challenging to get the signature from a VIP, right? Because they're busy. Let's look at this example. This is a collaborative study that we conducted with Professor Steinmetz, and she presents tomorrow morning, so please attend her presentation to learn about her particles. Here I will focus on methodology. Then we conducted a study for Nicole. We looked at cytokines in response to her particles, and we noticed that one of them induces IP10. IP10 stands for interferon gamma inducible protein, but we don't see interferon gamma. Why is that? To answer this question, we had to look at the principle of underlying methodology that we are using. Then we use ELISA. We analyze cytokines in supernatants collected 24 hours after incubation with the cells. During these 24 hours, many things may happen, which includes cytokine degradation or binding to receptors on the cell surface, which we subsequently remove the cells during centrifugation step. Then we prepare supernatants for analysis. If, however, we use orthogonal methodology, ELISpot in this case, it allows us to capture cytokine in real time as cells keep producing it. And so, indeed, then we looked at Nicole's particles using ELISpot. We detected low levels of interferon gamma, which confirmed to us that IP10, which we saw by ELISA, is not an artifact, is a real thing, just ELISA cannot detect it. Now let's look at the case studies to see how different models allow us to answer specific questions about nanoparticle immunotoxicity. When I talk about in vitro systems, I would like to talk about palmar-plantar erythrodysesthesia. It is immune-mediated inflammatory disorder of the skin, which is commonly observed in chemotherapy. And here on the slide, you see the list of the drugs. Among them are doxorubicin and pegylated liposomal doxorubicin formulation, Doxil. The problem is animals do not develop PPE. So how do we study this toxicity? We collaborated with Ann Dickinson and used autologous skin PBMC co-culture, and we demonstrated both by skin histology and cytokine analysis that we can detect inflammatory damage to the skin in response to doxorubicin and Doxil, similar to how this occurs in vivo. So this is an example when in vitro system can help us answering biological question which we cannot address using in vivo model. But there are situations where we have to look at animals. And one of these examples is from our collaborative study with Curadigm. Julie presents later in this session, so please attend her presentation to learn more about Curadigm product. Here I'll focus on methodology. So Curadigm has a nanoprimer, which is a nanotechnology concept intended to specifically and transiently occupy mononuclear phagocytic system to improve drug delivery. At NCL, we wanted to answer a question, where in the MPS does the nanoprimer go? What you see from this slide is that we conducted a live tissue imaging study, and we found nanoprimer accumulation in red pulp area of the spleen, which is a home to macrophages, and it perfectly aligns with intended mechanism of action of the nanoprimer. You also know that spleen is the largest secondary lymphoid organ, so we asked, can nanoprimer, in addition to improving drug delivery, help with vaccine? We conducted in vivo efficacy study, and indeed we demonstrated that administration of nanoprimer 30 minutes before administration of the vaccine improves both the magnitude and duration of the antigen-specific antibody response. I hope everyone in this room agrees with me that answering these two questions in vitro would not be possible. So what's beyond in vitro and in vivo? Here, I would like to use an example of our collaboration with Professor Afonin at the University of North Carolina in Charlotte. Professor Afonin and his team prepared a library of nanomaterials made of RNA and DNA or RNA-DNA hybrids, and my team at NCL analyzed their unique immunological signatures, which in this case include type 1 interferons. So what we found is that material type, RNA more than DNA, structure with globular particles being more immunostimulatory than the fibrous particles, size and connectivity contribute to the immunorecognition of these materials, but sequence complementarity didn't matter so much. Next, what we wanted to do, we wanted to see what if we can accumulate and connect physicochemical properties and immunological studies and use this data for machine learning to develop quantitative structure-activity relationship model. To achieve this, we reached out to Dr. Zakharov at the National Center for Advancing Translational Sciences at the NIH. Alex and his team used a random forest model, and they focused on one type 1 interferon, interferon beta, and came up with a QSAR model that uses molecular weight, melting temperature, and half-life of nucleic acid nanoparticles to predict the induction of interferon beta. So this is not enough. We made one step forward. Sankalp joined the team, and in addition to random forest, he used other algorithms, including popular these days transformer algorithm. He developed several modifications of this algorithm and demonstrated that transformer M2 has superior ability to predict not only interferon beta, but other types of type 1 interferon, alpha and omega, and type 3 interferon, lambda, based on the physicochemical properties. Based on this data, we created artificial immune cell or AI cell platform. It is publicly available free platform which researchers can use to look, use to input physicochemical properties of nucleic acid nanoparticles and predict interferon responses to these materials. So in conclusion, each nanotechnology product is unique and has its own immunological signature. Class-specific signatures exist. In my example, I referred to chemokine induction by lipid and polymer-based particles and interferon induction by nucleic acid nanoparticles. I also stated that nanoparticles behave as VIPs, which means that collecting their signatures is challenging and may require orthogonal methodologies. And here is the answer to the question that we asked in the beginning. What is ideal? Nothing is ideal. In vitro, in vivo, and in silico methods are all appropriate as long as they help answer specific questions and are scientifically justified. With this, I would like to thank my colleagues at the Nanotechnology Characterization Lab, our collaborators whose studies I used in my presentation, and thank you all for your attention. And thank you.